Start, sir.
Good morning, students. So, welcome to Botany class. This is Srikanth, a lecturer in Botany, SLB, TSRJC, Vikarabad. Okay, students. Today we are going to discussing about third unit. The unit name is genetics. So, in genetics topic, in genetics unit, we have only one chapter. The chapter is the principles of inheritance and variations. What is the chapter name? Principles of inheritance and variations. This is the chapter present in genetics unit. Okay, students. Now, let us observe what are the contents in genetics unit. So, we have some topics in genetics unit. First one is concept of genetics. What is the first one? Concept of genetics. And Gregor John Mendel and father of genetics. We have to discuss about Gregor John Mendel and Mendelian concepts. Terminologies of genetics. Advantages of pea plant. Mono and dihybrid cross. Test and back cross. Incomplete and codominance. Chromosomal theory of inheritance. And point mutation. These are the contents present in our topic. So, first one. We have to discuss about introduction of genetics. So, we commonly listen the word in our daily life in genetics. First, we have to know what is genetics. So, students, genetics is the young branch of biological science. This is a advanced, well-developed young branch in biology, that is a genetics. So, if you observe the introduction of a definition of genetics, the genetics is the study of heredity and variations. Study of heredity and variations. So, what is heredity? Heredity means the tendency of offsprings to resemble their parents. Okay, students, what is heredity? Some characters are transfers from parents to offsprings. Parents to offsprings. Those characters are called as inheritance characters. Such process is known as heredity. When we observe the offsprings are resemble their parents. That is known as heredity. Okay, so now what is variations? So variations means the offsprings are to vary from their parents. They're not as it is looks like their parents. Offsprings are must be vary from their parents, which is very essential for the evaluation. That is why the study which deals with these two terms, heredity and variations, the study is known as genetics. Genetics is a term first given by W. Bateson in the year 1905. Who given the word genetics? He was W. Bateson in the year 1905. Okay, so okay, students, here two pictures are there. If you observe carefully these two pictures, here, first picture, Sachin Tendulkar and his son Arjun Tendulkar. Here, the son is offspring. And there are some similarities are there between them and some variations are there. Because the Arjun Tendulkar gave inheritance characters from the Sachin Tendulkar. And also Arjun Tendulkar showing some variations from the Sachin Tendulkar. And whereas second picture is the KCR and his son KTR. Okay, here also some variations are there, some resemblance are there. So to study about these variations and heredity characters is known as genetics. What is that? To study about heredity and variations, known as genetics. And next one, now first we have to know about famous scientist in genetics topic. He was Gregor Johann Mendel. We have to learn about Gregor Johann Mendel. So he also popularly known as father of genetics. We already know about that. We already learned in first year about Gregor John Mendel. He was the father of genetics. He worked on Pisum sativum plant. Which plant? Pisum sativum. Its common name is garden pea. What is the common name of Pisum sativum? Garden pea. He published his experiments in the annual proceedings of the Natural History of Society Brunn in 1866. In the year 1866, he published his experiments in one proceedings. Its name was The Natural History of Society of Brunn in the year 1866. So, title of his publication. His publication title is Experiments in Pea Plant Hybridization. This is the title of his experiments. Experiments in pea plant hybridization. So this is a picture of Gregor John Mendel. This is a picture of garden pea. It is Pisum sativum. So now, later, Mendel died as unrecognized man. His studies remained dark for 34 years. Yes, it is true. Mendel died as unrecognized man. His studies remained dark for 34 years. Later, in the year 1900, three scientists independently rediscovered the Mendel's work. Those were Karl Korans, belongs to Germany, Hugo de Vries, belongs to Holland, and Erich von Schermack, belongs to Austria. So, Mendel findings were known as Mendelism or Mendelian laws of inheritance. So, these three members were rediscovered the Mendel's works. Later, they given the laws of Mendelism or Mendelian laws of inheritance. Okay, so who are they? Karl Korans, Hugo de Vries, and Erich von Schermack. Okay, now important terms. Now we have to learn about some important terms, especially in genetics. Before entering into the genetics topic, there are some exclusive terms are there in genetics, which are oftenly visible in genetics topic only. Before entering into the genetics topic, we have to learn some terms of genetics, okay, which is very important for that. Okay, so first term, what is the first term? Factor. In olden days, the heredity unit is called as factor. The factor name given by Mendel. In olden days, before the 19th century, the heredity unit is called as factor. This factor name given by Mendel. Later, the factor later called as gene. The factor later called as gene. The gene name coined by Wilhelm Johnson. Was there? Wilhelm Johnson. Wilhelm Johnson given the name of gene. And later, third term, allele. The allele is a term commonly visible in genetics topic. What is allele? Allele means a pair of contrasting characters are genes called allele. A pair of genes. So shortly called as a pair of genes called as allele. A pair of genes, that's it, enough. A pair of genes called as allele. Allele term given by Betson. Who given the allele term? Betson given the allele term. What is allele? Simple thing. A pair of genes. A pair of genes called as allele. Here some example. Capital T, one pair. Small t, one pair. That is why one allele. Capital T, one pair. That is why one allele. Capital Y. Okay. One pair, one allele. Small y, capital Y, small y. These are some allele example. So based on their properties, alleles are two types. Alleles are two types. Homozygous allele, heterozygous allele. What is homozygous allele? Homozygous allele means similar type of genes in allele known as homozygous. Homo means what? Homo means similar. Means in a pair, similar genes are present. See here, example, capital T, both are similar in nature. Small t, both are similar in nature. Capital Y, both are similar in nature. Small y, both are similar in nature. Which means similar type of genes present in pair, present in allele is known as homozygous. Heterozygous means a dissimilar type of genes in allele. What is heterozygous? A dissimilar. Hetero means, we know that hetero means a dissimilar or different, which means a dissimilar type of genes in allele is known as heterozygous allele. For example, capital T, both are dissimilar in nature. One is small, one is capital. Capital Y, small y, both are in dissimilar. Capital R, small r, both are in dissimilar. So these are the two types in alleles. Dominant factor and recessive factor. Dominant factor. So the gene of allele pair which can express itself. Dominant means expressing itself, expressing outside, which means the character which is visible to outside, known as dominant. The gene of allele pair which can express itself. In allele, two genes are present. Imagine these are the two genes. In these two genes, one gene is responsible to expressing character outside. One gene is responsible to not expressing to, unable to express itself. The gene which is expressing its character outside, that gene is called as, that factor is called as dominant factor. The gene of allele pair which unable to express itself. So the gene which is unable to express, which is visible to which is not visible to outside, is called as a recessive factor. Okay. For example, so here I given one example to you. So students, see here, what is dominant, what is recessive. So we can understand based on one example. Here, one example here. Here, one tall plant, one dwarf plant is there. They both are crossed, and tall will be produced in first generation. So the tall character is visible to outside. The tall character is expressing to outside. But dwarf character also present in this trait. Dwarf character is also present in this generation. But the dwarf character is not expressing outside. The tall character is expressing outside. That is why in this situation, the tall is dominant and dwarf is recessive. Okay, the dominant is nothing but the gene which is expressing outside, expressing character, visibility character. Okay, a recessive means invisibility character, unable to express character. That is dominant and recessive. Okay, next term, phenotype. Phenotype means external feature of organism or trait. So external feature means a physical appearance. Physical appearance of organism or trait is known as phenotype. Physical appearance means the visibility character. Means the character which is clearly visible to outside. That is external feature or physical appearance. That is known as phenotype. Genotype means genetic makeup of organism. Genetic makeup means where the genes are present internally. That is why the characters present internally in genetically known as genotype. Genotype means simply genetic makeup of organism. That is genotype. Okay, students. Now we know that Mendel selected pea plant. We already learned about Mendel experiments on garden pea, okay, the pea plant. So there are some lakhs, some millions of plants are there on the earth. Several plants are there on the earth. But why Mendel selected pea plant? Okay, why Mendel selected pea plant? Is there any particular reason behind in it? Is there any particular reason for that? Yes. So first we have to discuss about why Mendel selected pea plant. Is there any advantages of pea plant? So now let us see what are the advantages of pea plant. Advantages of pea plant. So why the Mendel selected pea plant? Because the pea plant is showing some special characters, some advantages. That is why Mendel selected pea plant. What are those advantages? First one, annual plant. It can be grown and crossed easily. Annual plant means, what is mean by annual plant? Which complete its life cycle within a year. What is annual plant? Annual plant means, annual means 1 year. Means the plant which complete its life cycle in a year. Annual plant. The pea plant is the annual plant. It can be grown and crossed easily. The pea plant is growing and crossing very easily. Okay. And pea plant has bisexual flower. Pea plant has bisexual flower because the single flower has stamen and carpellar pistil. Same flower has male and female reproductive parts. That is why the pea plant has male and female reproductive part in single flower. That is why bisexual flower is present. Flower shows self-pollination. So self-pollination means the stamens, the pollen grains are reaches to the same flower stigma. That is self-pollination. So in pea plant, sometimes easily self-pollinations are takes place, and sometimes cross-pollinations are takes place. And pea plant showing well-defined, excellent characters. And it has a short life cycle and produce large number of offsprings, number of plants within a short period. Because annual plant. The pea plant is the annual plant. That is why it has short life cycle and produce large number of offsprings. Produce large number of offsprings. That is why so Mendel selected pea plant. These are some special character showing by the pea plant. That is why Gregor John Mendel selected pea plant. So this is about information of pea plant. So garden pea plant. So its scientific name is Pisum sativum. The Pisum sativum belongs to the family of Fabaceae. Number of chromosomes in Pisum sativum is 7 pairs, which means 14 chromosomes. Which means 14 chromosomes or 7 pairs. So here Mendel selected 14 true breeding traits, which means 7 sets of characters studied by Gregor John Mendel for his experiments For Gregor John Mendel experiments, Gregor John Mendel selected 7 pairs of characters, which means 14 true breeding characters. 14 true breeding characters. Those are seed shape, seed color, pod shape, pod color, flower color, flower location, plant size. Okay, seed shape, round and wrinkled. Seed color, yellow and green. Pod shape, inflated and constricted. Pod color, yellow, green and yellow. Flower color, purple and white. Flower location, excellent terminal. Plant size, tall and short. So these are the 7 pairs of characters observed by Gregor John Mendel. So by this, Mendel conducted artificial cross-pollination experiments using true breeding plants. So in these true breeding plants, Mendel conducted artificial cross-pollination. Artificial cross-pollination using these true breeding pea plants. Mendel conducted hybridization experiments on garden pea for 7 years. How many years? 7 years. Mendel conducted hybridization experiments on garden pea for 7 years between 1856 to 1863. 1856 to 1863. In this year, Mendel conducted hybridization experiments on pea plant. Based on his experiments, Mendel proposed 3 inheritance laws. 3 inheritance laws. Those are law of dominance, law of segregation, law of independent ascertainment. Okay, how many laws are there, Amma? So, 3 laws: law of dominance, law of segregation, law of independent ascertainment. Who proposed these 3 laws? Mendel. These 3 laws also known as inheritance laws. We explaining about law of dominance. Mendel choosing monohybrid cross. So explaining about law of dominance, Mendel choosing monohybrid cross. To explaining about law of segregation, Mendel choosing monohybrid cross. Same. To explaining about law of independent ascertainment, Mendel chooses dihybrid cross. Okay, how many laws are there? 3 laws are there. Okay, so these 3 laws are proposed by Gregor John Mendel, conducted hybridization experiments on garden pea plant for 7 years. How many characters he observed? How many sets of characters he observed in pea plant? 7 sets of characters. 7 pairs of characters he observed in pea plant. Okay, so these are the 7. These seven sets are known as pure breeding traits. What are that? Pure breeding traits. Pure. What is meant by pure breeding? Pure breeding traits means having undergone continuation self-pollination. What is pure breeding? Having undergone continuation self-pollination. Continuation self-pollination for several years. Having undergone continuation self-pollination for several years. And shows the stable trait inheritance and expression for several generations. What is true breeding? Having undergone continuation self-pollination, shows the stable trait inheritance and expression for several generations. Which means to expressing the desirable characters for several generations. So Mendel undergone self-pollination in pea plant. That self-pollination process is known as true breeding. So these 7 sets of characters, these 14 pairs of 14 characters plants are known as true breeding traits. This 14 here 14 pure breeding plants are here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 14 pure breeding plants here, and with 7 pairs, and experiments experimented on pea plant of these 14 pure true breeding plants, and proposed 3 inheritance laws. Those laws are known as Mendel inheritance laws, law of dominance, law of segregation, law of independent ascertainment. Most important among these 3 laws, very, very important: law of dominance, law of segregation, law of independent ascertainment. So explaining about law of dominance, Mendel chose a monohybrid cross, and the same law of segregation, monohybrid cross. Law of independent ascertainment, dihybrid cross. Now we will discuss about law of dominance. What is that? Law of dominance. Let us see what is law of dominance. Okay. See, students. Law of dominance. Law of dominance means what? Any one of parental character expressed in next generation. Where? In next generation. Any one of parental character expressed in next generation is known as law of dominance. So, what class he taken for the explaining about law of dominance? Mendel chose monohybrid class. What is monohybrid class? Monohybrid class means cross between one pair of character. Class between one pair of character is known as monohybrid class. How many pairs of characters observed by Gregor John Mendel? 7 pairs of characters. Among those 7, he taken one pair of character for monohybrid class. Okay. So for the explaining about law of dominance, so let us take example: tall plant crossed with dwarf pea plant. True breeding tall plant crossed with true breeding dwarf pea plant. Okay, what happened? See, so in F1 generation, F1 means first filial generation. What is F1? First filial. Okay, first filial generation. First filial generation. F1 means first filial generation, first generation plant that tall will be produced. Is there any tall in parents? Yes. So what is law of dominance? Any one of parental character expressed in next generation. Yes, as usual, tall will be produced. Tall is there in parents. That is why law of dominance satisfied here. Okay, law of dominance satisfied. That is, law of dominance means in next generation, either tall or dwarf will be produced. Any one of parental character. In next generation, what will be produced here? Tall will be produced. That is F1 generation. So here, self-pollinated in tall plants. He self-pollinated in tall plants. So if he self-pollinated the tall plants, so second generation plants will be produced. That is known as F2 generation plants. Means second filial generation, which means second filial generation. In second filial generation, if we self-pollinated the F1 generation tall plant, in second filial generation, 75% tall will be produced, 75% tall will be produced, and 35% dwarf will be produced. Okay, sorry, 25%. Okay, how much? 25% dwarf will be produced. Okay, correct that point here. 25% dwarf and 75% tall will be produced. Okay, so here tall and dwarf, both characters are present in parents. See, is there any other character apart from tall and dwarf, other than the tall and dwarf? No other character. No other character in next generation. In next generation, whatever it may be, F1 generation or F2 generation, whatever it may be, in next generation, tall or dwarf, any one character developed. In F1 generation, tall will be produced. Tall character is present in parents. In F2 generation, tall and dwarf both are present. Tall and dwarf both are present in F2 generation, present in parents. Okay, students. So, to understanding about monohybrid cross, to understanding about law of dominance very easily. So, we have to take symbolic method. Okay, to understanding about more easily of law of dominance or monohybrid cross, we have to take symbolic method. So here, capital letter indicating about dominating character, small letter indicate about recessive character. Most important, these two are. If there is any capital letter that is dominant character, is there any small letter that is recessive character? Yes. By using the symbolic method, we have to fill the Punnett square. What is Punnett square? Punnett square is nothing but the Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or breeding. In a cross in a breed, some traits, some offsprings are produced or developed. To filling these traits or genotypes, or to filling these traits genotypes, we have to use the one square box. The square box is called as Punnett square. Okay, what is Punnett square? To fill the genotypes of offsprings. To fill the genotypes of offsprings, Reginald C. Punnett. Okay, Reginald C. Punnett, he given the Punnett square first time. Okay, so by using the Punnett square, we can determine the probability of offspring and genotype of offspring. So we can easily find out the probability and genotype of offsprings in a cross, in a breeding, in a cross. We can easily find out by using the Punnett square the offsprings genotypes and offsprings probability in generations. Okay, so now let us introduce the symbolic method. See, students, here monohybrid class. See, first, pure homozygous tall pea plant. Why it is called homozygous? Because two genes are same. Two genes are same. Capital T, capital T. Pure homozygous tall pea plant crossing with pure homozygous dwarf pea plant. Okay, small t, small t. These two are parents. Okay. So they are giving gametes. From the capital T, one gamete capital T. From small t, one gamete small t. Both are crossing together. F1 plant will be produced. F1 generation plant. Okay. The F1 generation plant, what is that? Capital T, small t. Capital T, small t. See, students. So if you observe this capital T, small t, here one capital letter is present, one small letter is present. What I said, the capital letter always indicating about dominating factor, and small letter indicating about recessive factor. So here capital letter is present, that is why the tall. Small letter also present, that is why dwarf. What is the dominant factor? Tall. That is why, what is the phenotype of this F1 plant? Yes, tall, because dominating one is capital. That is why the capital T is dominating to small t. That is why the tall character is dominating to dwarf plant. That is why the dominating character is tall. That is why the phenotype of type is tall. The tall plant will be produced in F1 generation. What plant is produced? The tall plant is produced in F1 generation. Okay, so okay, students. So he self-pollinating the tall plant which is developed in F1 generation. Okay, so what he did? He did a self-pollination the plant which is produced in F1 generation. So what proportions are developed in next generation? That is F2 generation. In F2 generation, 4 types of plants will be produced. Okay, so here this is a Punnett square. These are the gamete, capital T, small t, capital T, small t. So horizontal and longitudinal. These are horizontal rows. These are longitudinal rows. This is a self-pollinating. That is why same flower pollen grains are reaches to the the pollen grains are reaches to the same flower stigma. That is why horizontal rows and longitudinal rows gametes are both are same because the flower is same. That is why. Okay. So, but here the flower is different. That is why if we fill these when in Punnett square, all capital T, small t traits will be produced in F1 generation. That is why we can take only one capital T, small t in F1 generation. Here, selfing means the pollen grains are reaching to the same flower stigma. That is why selfing. That is why he taken horizontal rows and longitudinal rows gametes are same. Capital T, small t, and capital T, small t. Both are same. Small t and capital T both are same. That is why if we fill the box, capital T. So capital T, capital T, small t. So capital T, small t. Capital T, small t. So capital T, small t. Small t, small t. Small t, small t. Okay. So capital T, capital T, small t. So capital T, small t, small t, small t. Okay. So okay, students. So here, first one. What is the first one? Pure or homozygous tall. What is the first one? Pure or homozygous tall. Second one, capital T, small t, hybrid. This is a hybrid because both dissimilar genes. That is why this is hybrid. So hybrid heterozygous tall. Heterozygous tall because the both are dissimilar. This is also capital T, small t, both. This is also same hybrid or heterozygous tall. Small t, pure homozygous dwarf. Pure homozygous dwarf. So let us find out the ratios. So now, what is the phenotype ratio? How many talls here? Capital T. This is also tall. Capital T, small t. This is also tall. Capital T, small t. This is also tall. Small t. This is dwarf. How many talls here? 3 talls here. How many dwarfs here? 1 dwarf here. 1 dwarf is here. That is why the external appearance character known as phenotype. The phenotypic ratio is tall is to dwarf. Tall is to dwarf. That is equal to how much? How much? The tall is to dwarf, that is equal to how much? 3:1. Simply 3:1. See, phenotype ratio is equal to tall is to dwarf, that is equal to 3:1. So why it is called 3:1? Because here in this box, the 75% are tall will be produced. This is tall, this is tall, this is tall. Okay. Yes, this is tall, this is tall, this is tall, this is tall. Both these three are tall, but the genotype is different. We don't want that genotype now. We want only now phenotype. So capital T, capital T, pure tall, capital T, small t, hybrid tall, capital T, small t, hybrid tall. So these three are tall. 75% is tall. Remaining 25% dwarf. Small t is dwarf. 75 is to 25. That is equal least 3 is to 1. Nothing but 75 is to 25. Nothing but 25 ones are 25 threes are 3 is to 1. Nothing but last to final ratio is 3 is to 1. Our ratio is phenotype ratio is in monohybrid cross 3 is to 1. Now let us find out a genotype ratio. Let us find out genotype ratio in F1 generation. So genotype ratio. Capital T, tall. Capital T, small t, tall. But these both talls are different. The capital T is the homozygous tall. Capital T, small t is the heterozygous tall. So how many homozygous tall here? 1. How many heterozygous tall here? Is 2. 2. What is the last one? Small t, small t. Small t means homozygous or heterozygous? Yes, homozygous. Homozygous, taller dwarf. Yes, dwarf, that is equal is to 1. Here, this is the genotype ratio of monohybrid cross between the tall and dwarf. Means pure homozygous tall is to homozygous tall is to heterozygous tall is to homozygous dwarf. How many homozygous tall? 1. How many heterozygous tall? 2. How many homozygous dwarf? 1. It is equal to 1 is to 2 is to 1. See, genotypic ratio homozygous tall 1, heterozygous tall 2, homozygous dwarf 1. It is equal to 1 is to 2 is to 1. 1 is to 2 is to 1. So probability. We can find out probability also, which is very important for NEET and MCET purpose. So we have to find the probability. Out of 4, how many threes are there? How many talls are there? Out of 4, how many talls are there? 1, 2, 3. 3 talls are there. Tall probability. What is the tall probability? 3 by 4 or 75% or 0.75. This is the tall probability. What is the dwarf probability? What is the dwarf probability? Out of 4, how many dwarfs are here? 1 dwarf. That is why 1 by 4 percentage. Out of 100 percentage, how much of dwarf here? 25%. That is why 25% and 0.25. This is the probability of tall and dwarf phenotype probability. If they ask for the what is the probability of the homozygous tall? What is the probability of the heterozygous tall? What is the probability of the homozygous dwarf? This is about that. In out of 4, how many homozygous tall here? In out of 4, how many homozygous tall? Yes, 1. Yes, only 1 out of 4, only 1 homozygous tall here. So 1 by 4. Out of 100, how much of homozygous tall here? Yes, 25%. So 25%. Nothing but 0.25. Let us take heterozygous tall. So out of 4, how many heterozygous tall here? Yes, 2. That is why 2 by 4. So in 100%, how much of heterozygous tall here? Yes, 50%. Very good, 50%. That is nothing but 0.50. Homozygous dwarf. So out of 4, how many homozygous dwarf here? Yes, 1. That is why 1 by 4. In percentage, how much? How much? 25 percentage. Nothing but 0.25. Nothing but 0.25. This is about probability and phenotype ratio and genotype ratio. So there are two crosses are there. So sometimes normal cross, sometimes reciprocal cross. What is normal cross? What is reciprocal cross? So first, normal cross, making cross between female tall plant, tall parent, and male dwarf parent is known as normal cross. Making cross between female tall parent and male dwarf parent is known as normal cross. See, what is that? So we have to take this parent is the female. So this parent is the male. So to making male and female parents from the tall plant, we have to cut the anthers or stamens. That means emasculation. So and from the dwarf parent, we have to cut the what one? And gynoecium. From here, we have to cut the stamen. From here, we have to cut the pistil or carpel. Then this is changed into female parent. Now this is changed into male parent. So if we cross between these two parents, that is known as normal cross. So if we cross the opposite direction, making cross between the male tall parent and female dwarf parent is known as a reciprocal cross. What is reciprocal cross? Making cross between male tall parent and female dwarf parent is known as reciprocal cross. So what is reciprocal cross? We have to take this tall plant as male parent, dwarf plant as female parent. So if we cross between them, that is known as reciprocal cross. So this is about normal cross and reciprocal cross. Okay. So now, so here some model and NEET and MCET questions are here for on monohybrid cross. Okay, students. Here some model. I given one model NEET and MCET question on monohybrid cross. Okay, student. See this question. In garden P, the flowers may be axial or terminal. Axial indicated by the capital A, terminal indicated by the small a. Okay. So in position, what proportion of the offspring in the following crosses would be expected be axial? So these are the questions. First question, capital A small a into capital A small a, capital A into capital A small a. First one, capital A small a, capital A small a. So if we divide into gametes, capital A small a, capital A small a. First one, capital A, capital A. First one, capital A, which will be produced capital A. Second one, capital A small a. So what will be produced? Capital A small a. Third one, capital A small a. Okay, now capital A small a. So capital A small a. So from here, okay, and last one, small a, small a, small a, small a. Okay, student, capital A. What is the capital letter property? Always the capital letter indicating about dominant property. Okay, so capital A, capital A, nothing but pure axial. Capital A, small a, nothing but capital letter is present. That is why which is axial. Capital A, small a, this is also axial. Small a, small a, there is no any capital letter. That is why it is terminal. So axial flower plants are 3/4 or 75% or 0.75. Out of 4, how many axials are there? 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 3 axials are there. That is why 3/4. Or out of 100%, how much of axials percentage? 75%. Nothing but 75. So in first situation, what is the axial proportion? What is the axial probability? 3 by 4 or 75 or 0.75. So now, okay students, take this screenshot and do second question at your home. This is your homework. Okay. So you have to find out the axial proportion in second question. So here, this is the first question solution. Yes, by using this first question solution, so you have to find out the second question answer. In second question, what is the axial percentage? You have to find out at home. This is your homework. So next, second question. So this is a one type of question commonly asking in NEET and MCET purpose. And in a cross between a yellow and green, in a cross between a yellow and green seed pea plants, all F1 members are yellow. But F2 generation raised by crossing two such F1 consist of approximately 75% yellow and 25% green seed pea plants. So what will be the offsprings be like if two F2 greens mated? What will be the genotypic ratio in the population of yellow seeded pea plants in F2 generation? Okay, students. So first of all, we have to read the question, understand the question carefully. What he given in a cross between yellow and green seeded pea plants? Okay, he crossed between yellow seeded pea plant with green seeded pea plant. All F1 members are yellow. Ultimately, all F1 members are yellow. Okay, this is so yellow seed pea plant crossing with green seed pea plant. What happened in F1 generation? All F1 members are yellow. Yes, okay. So, but F2 generation raised by crossing two such F1 consist of approximately 75% yellow and 25% green seed pea plants. Automatically, so he self-pollinated the F1 plant, and in F2 generation, what happened? 75%. 75% what will be produced? 75%. Yes, 75% yellow seed plants produced. 25% green seed pea plants produced. Okay, so this is a sentence. This is a one sentence, one class. So now, by using this class, by using this sentence, now we have to find out the question A and question B. We have to find out the question A and question B. So simple thing. What is the question A? What will be the offspring be like if two F2 greens mated అన్నాడు. Okay, so F2 greens.
Srikanth sir, time complete time sir, time up.
Okay.
12:30, 7 అయింది.
Sir, completed, sir?
Completed.
Thank you, sir.